few disclaimers. So I am an advisor, or Farsight. I'm a member of the CiPA steering committee, as Thomas said, as well as the co-chair of the Ion Channel Working Group. And really, what I'm going to tell you now, my views, are really my views and not necessarily those of the company or any of the groups that I'm affiliated with. So really, as the first speaker, I'm kind of just setting the tone for this, the talks that are going to come. But what I'd like to do is, there's been a lot of CiPA speculations, and there's a lot of CiPA presentations, and I understand all that, and I think it's quite exciting. But I think I'd like to take this time to kind of bring us back to what CiPA is all about and what it's likely going to do. And I think there are different ways to view the CiPA initiative and the potential impact of CiPA. I'm kind of the half full glass kind of guy, so I like to have kind of a positive view on things, and I'm quite excited actually about this effort because it's the normal progression of all the work that we've been doing over the past decade actually with HERG. And I think it's quite time for us to move to the next step, and really the next step is to move to pro-arrhythmia. So we're all involved in doing work, and I think that we've done a fantastic job at identifying, HERG blockers and trying to define, the potential impact on the clinical outcome. And TQTs have been put in place and provide really great information about QT prolongation. But we're at a point now where QT prolongation doesn't really tell us whether or not a drug is good or bad. So really the goal of CiPA is understand the pro-arrhythmic potential of drugs before they go in the clinic. So TQT studies are typically performed phase 2 on healthy volunteers, and you still get some surprises, but what's probably more challenging is not the surprises, but the in-betweens, and that's when you don't quite know what to do next. So hopefully CiPA is going to allow us to identify really early on, compounds that have potential pro-arrhythmic activity, and that's going to lead to increase the efficiency of the development pathway. So help us move, the compounds forward. There's also an impact that is not talked about, but that could be quite significant, and that is the effect on labeling. So we have a bunch of drugs that have a QT label on them, and it affects, of course, the sales of the compounds and the use of the compounds. And part of the hope with CiPA is that we are able to take compounds that have a QT label, run them through the CiPA assays, and change that label. And if that were to happen, then we would recover quite a few compounds actually that are known to prolong the QT, but their pro-arrhythmic liability is somewhat confusing at this point. So, the impact again will be hopefully increased compound in development. And finally, what's really significant is it'll lead to the revision of the S7B guidelines as well as the E14, and in fact eliminate potentially E14. So no more need for a specified or a defined TQT study, and revision of the S7B guideline. And the way I view it is we've been using S7B, so S7B has been out since 2005, so it's been a decade now that we've used S7B. And the way that it came about, and believe it or not, I was part of the original DRU-SAFE committee that drafted the ion channel portion of the S7B, and we knew doing it that it was really giving the choice to people. And what it turned out to be is to create a situation where now everybody has their own assay, their own solutions, their own cell lines, their own protocols. And it's kind of a mess out there. What CiPA is doing is we're trying to do that ahead of time. So the idea is to come up with protocols that are standardized, solutions that are standardized, so that everybody starts out on the same footing. Of course, probably things are going to change, so people are going to make some changes internally. But for a submission package, there's going to be well-defined conditions that everybody will have to adhere to. And that's really going to level the playing field. So again, I think one big advantage over S7B is that a company like Pfizer has a lot of resources. Biotech companies don't have access to as many resources as we do. So to some extent, we have a competitive advantage from the get-go. And CiPA is going to make sure that it's leveling the playing field, because ultimately protocol solutions will be available to all. And as well as the in silico model that is used in CiPA is going to be available to all. So again, I think a very important thing will be putting everybody on the same footing. So what are the issues with the current preclinical paradigm? I mean, I think we've done really, really well at setting up assays that allow us to identify hERG blockers. We've done quite well, actually, in using these numbers and setting margins of safety within each of our own companies and making decisions on whether or not to move compounds forward based on these margins. So what's the problem? Well, the problem is always the same. So we can do all that, and we can move compounds in the clinic, and we can expect these compounds to not cause prolongation, because you're not going to see torsade in a TQT study. But then again, we find ourselves with compounds that we've well defined what the prolongation is, so the thorough QT, so the corrected QT, what that prolongation is, but we still don't know if that's good or bad. And it's easy for a compound that prolongs the QT, by 50 milliseconds, but then you would say that your preclinical strategies would screen those out. The difficulty is when you get to a TQT study and you have 12 millisecond prolongation. So you move it forward, you don't, and it's then it becomes a risk-benefit decision. But sometimes it's not always. They're not all cancer drugs, and even cancer drugs, given the longevity of life now on many of these drugs, it's not a slam dunk going in with a compound that has QT effects, even if it's an oncology drug. So the issues are, we know it's a hERG blocker, we know it prolongs the QT, we know about how much, we still don't know if it's proarrhythmic or not. So the goal of CiPA is really to change that. We focus quite a bit on hERG, and it's an important player. But again, as I state here, and as you all know, not all QT prolongation represents an effect on hERG. Not all hERG blockers are proarrhythmic, so we know that. Some hERG blockers actually are antiarrhythmics. Now, very few of them. And there's always the poster boy. So verapamil is always the poster boy for a hERG blocker that doesn't prolong the QT and doesn't cause torsade. And there are very few of those, but they do exist. And of course, we would miss them or potentially discard them by looking at hERG only. And really, we can identify QT positives, but again, we can't really say if they're proarrhythmic. So then the TQT is was a good idea in trying to define with some precision the amount of prolongation that you get from a compound. And it plays an important role presently in labeling, but I guess in some cases has somewhat of a negative impact on labeling. Now the methodologies are well defined, so pretty much all the companies do it pretty much the same way. You need a positive control, so most people use moxifloxacin. It's a very well-behaved compound. So as an approach, TQTs are good because, again, you can define usually, and with a compound that has a nice PK, you can define pretty well the amount of prolongation that you're going to get. Of course, again, and I was alive and contributed then. When the E14 document was getting written, it was slightly with a delay from the S7B, but there was no real communication between the clinicians and the preclinical folks. And so nowadays, there's very limited interest in the preclinical data. Now that's not completely true. So I would say that depending on the companies that you are, some of the clinicians will put some emphasis on the preclinical. I think that it's a blanket statement, but I think that overall in Europe, the EMEA do put a lot more emphasis on the preclinical than the FDA, for example. So I think it's important. But it remains that a TQT studies is about $2 million US. Pfizer, that has a very large portfolio, spends a tremendous amount of dollars. And in fact, in some situations, we have to shift money from preclinical to clinical because of the cost. Now, I'm not saying that it's the TQT is the most expensive part of it, but it is expensive. And when you look at 40 compounds in a year in the clinic, it gets to be a pretty important expense. And the reality is that, again, we can quantify QT prolongation, but then you're stuck with making a decision on whether or not you push it forward. And there will always be those that are no-brainers, right? So you'll have those that will have a dramatic effect on the QT, and those that will have less than 5 milliseconds on the QT. And then you have all those in the middle, and it becomes somewhat of a difficult decision process to, especially when you have a large portfolio moving quite a few compounds forward, make a decisions on which one are important. So what happens? Well, from a preclinical point of view, there is still this, the regulators are still very careful, and it makes sense, but the data is telling us now that we have to move to something else. The regulators are still very careful, and anything below 10 milliseconds, at least with the FDA, they're quite reasonable. It's not necessarily the case for every regulators, but nonetheless, small effects on hERG and small QT prolongations still remain quite an issue. And that definitely leads to termination of drugs, the emphasis on candidates, the emphasis on targets, improper discontinuation of programs, labeling that will definitely affect the availability to patients. And you can be sarcastic and say, well, it's all a question of money for the companies, right? So ultimately, they're trying to save money, and that's true. I mean, money and budgets and all that is all very important. But in the end, and in reality, we are putting things away, or throwing away compounds that are pretty effective, and that are pretty effective against the target. And you just, the risk-benefit balance is not just appropriate, but ultimately, it's the patients that don't have drugs to treat their conditions. And so it's quite important. So CiPA is this for there's a risk that people may be confused now because there's so much CiPA everything going on, but CiPA remains two components. On the left hand, you have ion channel data that will be incorporated in an in silico model. And on the right-hand panel, you have integration of this data in stem cells and looking for either confirmation of what you'll find in the in silico model, or even find things that were not observed in silico because all you were doing was inputting ion channel data in there. So it's got limitations. There's no doubt about it. Anything that doesn't have to do with ion channels and still prolongs the QT, and that could be, central nervous system effects, effects on blood pressure, effects on heart rates. I mean, many different things. It's not going to catch. But that's not the purpose. The purpose of this is not to, with the first go around, catch everything, be, perfect, and walk away with turnkey assays that are going to allow you to develop everything and anything that affects her. But it's a first step. It's a first step in the direction of really taking compounds that have some hERG activity and really putting an evaluation on them that will allow you to, clearly move them forward with a known risk, not of QT prolongation, but of proarrhythmia. So my title says, I don't know if you remember my title, but because I'm not sure I do, but it says basically, CiPA is going to revolutionize things. And when you think about it, you can ask, well, what's the big revolution? So let's take a look at what CiPA is going to do, because it's going to happen. I mean, whether you agree with it or not, whether you agree with the current version of it or not, it is the next step. We have to go there. We have to go to proarrhythmia. And so the first thing that it's going to do is really standardize the ion channel assays. And the work that is being done initially is all done by hand. So it's manual voltage clamp, and the purpose being, there's a poster 19, I believe, talks about voltage clamp, manual voltage clamp still being the gold standard. That's kind of the idea. So the work is being done manually, but ultimately things are moving to high throughput systems, and it's going to standardize the way that the assays are run on the HT system. And so imagine that. You read a paper and everybody is using the same protocols and the same solution. How boring is that going to be? But it's going to level the playing field. So what this is going to lead to is, prevent attrition due to testing for hERG liability only. And then you're going to move into an in silico model. And the in silico model is going to be available online. And it's going to be the same model that everybody is going to use. And you will use the data that you generate in-house. And it can be the data that you generate using the CiPA protocols or any other protocols or any other data that you generate. Because, companies and we at Pfizer will continue to have assays that we will run independently. CiPA is all about for internal decision. So you're going to be using, for the CiPA purpose, an in silico model that is going to be the same for everybody. So the model will be parameterized such that by inputting your information, you'll be able to get information on whether or not your drug, even though it's a hERG blocker, is proarrhythmic or not. And you'll do that from your desk. So you're not going to need supercomputers. Just your little PC on your desk is going to be sufficient. So that's really going to give you an index of proarrhythmia, and that's what we're all looking for. And then you will check things in stem cells. Now, you can, again, at this point of time, there are 1.5 million posters on stem cells upstairs. And you can believe or not in stem cells, but that too is going to evolve, and it's going to evolve to the point of where you would like to see the stem cells. And it may not be this year or next year, but it's likely going to happen. And CiPA is going to establish best practice. So again, you can use them any way you want internally, but for the CiPA package, it's going to be established, well-defined, and so everybody on the same playing field. So that's going to provide a more complete assessment of the effects on human cardiac electrophysiology. It's not going to be perfect, but it's going to be a different assay than just looking at a single channel. And again, it's going to give you the advantage of either reproducing what you see in silico or picking up things that will make you go, well, did we miss something, and run it in other assays. The whole idea is to focus on proarrhythmia rather than QT prolongation. So, a shift in the way that we're thinking. It is going to replace the TQT studies. I mean, there's no doubt. So there's a paper by Borj Darpo that's already out in December of last year, where they did a small number of compounds, a more solid phase 1 study, and it's going to happen. And replacement of the TQT study is likely going to happen before we come in with the final plans for the in vitro and in silico. But that too is unstoppable at this point. And all of this, all of these approaches combined are really going to allow us as companies to move things forward in areas and for diseases that right now we are stuck. And therefore, it's going to allow new drugs to reach patients and really answer an unmet medical need, which for some diseases is so needed, you wouldn't believe it. And so I think, ultimately CiPA is going to be a winner. And I hope that I've convinced you that, at least it's a good thing. There's a lot of effort, there's a lot of presentations, there's a lot of CiPA approved data out there. The way I see it is, people are working, generating interesting data. Ultimately, the package is going to come from the CiPA group. But I see the interest as something very positive, and I'm very excited about this. And it's something that I feel is really needed, and hopefully I've convinced you that it's the case. So thank you.
So